Hello, my name is Anthony Cucci, and I am the Fraternal Relation and Development Associate at the Masonic Medical Research Institute. The following presentation took place on October 6, 2020, and the content of the conversation was related to our current coronavirus research efforts. The participants of this conversation were the Most Worshipful William M. Sardone, Grand Master of Masons in the state of New York, our Director of Research and Executive Director, Dr. Maria Contreras, and two of her primary investigators, Drs. Nathan Tucker and Sathya Unidarthi. Thank you and enjoy the presentation.
We are going to now start off for the evening here, and I want to thank everybody for joining us. My name is Most Worshipful Bill Sardone, and I'm the Grand Master of Masons in the state of New York. And I reintroduce myself only because of the fact that we have a lot of individuals who join this call who are not necessarily from New York and not necessarily from the Masons. And it is my pleasure and my honor to be here this evening and to introduce to you, some of you, but most of you probably already know, Dr. Contreras from our Masonic Medical Research Institute. But before I go on, let me just tell everybody that we already have people on from around the world on this call. Nadia in Bulgaria, I see her out there. I see her waving at me. We have people from South Carolina, Lebanon, Brazil. We have people as far away as Hoboken, New Jersey. I don't know how we got that far, but we did. And we have individuals that are still continuing to log on here to be with us. It is a pleasure for me to have the acquaintance and the opportunity to link with the individuals who are not only on this particular Zoom, but all of the staff and scientists that are associated with the Masonic Medical Research Institute. Obviously, Dr. Contreras is a catalyst in doing new things. And they have many research balls in the air, but right now the word COVID is on the tip of everybody's tongue. And Dr. Contreras, I just want to tell you one thing that you probably may not be aware of because you're basically a shy individual, and that is that you are a role model. And that happened, came to my knowledge, not that I didn't know it before, about 2 or 3 nights ago when I had a dedication of windows that we had in a lobby of 23rd Street in Grand Lodge, where I dedicated windows to some of our Masonic youth groups, the girls and the boys, or the young men and the young ladies. And usually when they come into the building and we're ready to do something, they're oohing and eyeing at me and my apron and my top hat, and then they saw a picture of you, and that was it. I was gone. They lost. They just went right over to your picture and said, "There's my role model," because you spoke on one of their Zoom calls, I think for Triangle. And after that, they wanted nothing to do with me except for when I was bringing the pizza in later on. So you are a role model for young men and young ladies alike, but the young ladies in particular really, really, really look up to you. And your picture is in one of the displays that we have of the Masonic Medical Research Institute in one of the windows. We have it set up there, and Anthony Cucci is working on getting me some new pictures to put in there. But when they saw your picture, they didn't know I existed anymore. So it was fine. It was a great night. They were happy. But you are a role model, and I know that each of you is a role model in your own way for what you do. So this time, it is my pleasure and my honor to turn this over to Dr. Contreras. And you can each, if you want to, put a question into the Q&A portion at the bottom of your Zoom screen or wherever it may be. We have some people putting it into chat. Let's try and keep it all in one place if we can. It'd be nice if we could. So let's see if we can move on here. Dr. Contreras, thank you so much.
Thank you, Grandmaster Sardone. It's a pleasure to be here with you tonight. Thank you for the kind words. I hope that I can pass it forward or play it forward and inspire some young people like I had been inspired in my life in moving forward in my career, my life, and in general to sort of methods of how to be successful and happy in your life, which I think are hand in hand. So if you do what you love, you tend to be happy. So I'm delighted to be here tonight. I'm going to introduce my colleagues who are here with me tonight. Dr. Sathya Unidarthi, who was recruited recently from the Ohio State University. did his postdoctoral fellowship there and is joining us here as a junior faculty here at MMRI, and Dr. Nate Tucker, who came from Mass General Hospital at Harvard University in Boston, just joined us as well in March. Both of these are my newest recruits, actually, and is currently working on areas associated with COVID directly, but has some both of them have cardiovascular interests as well. So I'm going to talk a little bit about MMRI, and they're going to share with you some of the work that they're doing directly with respect to COVID. So I'm just going to go ahead and share my screen now so that I can get on. Give me 1 second. Okay, can everyone see my screen?
I can.
Okay, great. So today we're going to talk a little bit about the impact of COVID-19 and how the Masonic Medical Research Institute is leading the way to fighting this disease with our research. So just a quick introduction about who we are. I was recruited here, I was fortunate enough to be recruited here in January of 2018. So I'm just completing now my second full year. That right? Third full year here at MMRI. And we've grown from about 17 people when I first got here and only about 7 scientists to over 50 people now with 35 scientists at the MMRI. And we've recruited individuals from across the nation, top scientists that are working at the peak of their game in cardiovascular research and in other areas of science that I'm going to talk about in a few minutes. So our growth since 2018 has been pretty substantial. If you haven't had a chance to visit us here in Utica, New York, I would encourage you all to please come next time you're traveling around the region. We've built a number of new spaces. We've renovated, almost completely renovated our entire building, second, third floor, and we just completed our basement renovation just this week. And we have only one wing of the Institute left to build and hope to achieve that sometime next year. We've been publishing in top-tier journals, we've recruited amazing scientists and a new administrative team, and we've bridged our academic network to collaborations within the nation and worldwide to our investigators here at the Masonic Medical Research Institute. And the most important piece of what we do is we depend very much on grants to do our research science, and we've been able to achieve receipt of multi-year grants from federal agencies, including the NIH, as well as state agencies as well. In terms of our mission and vision, our goal is to really improve the health and quality of life for all humankind. We want to conduct high quality biomedical and translatable research. We want to teach basic science as well as clinicians and students who can help perpetuate our research around the world and eradicate diseases worldwide. We want to understand the molecular mechanisms of disease and to develop cures that can be treatments for our ailments of tomorrow. We have various strengths at our research institute. These are some of the 4 core missions of the institute, which include genetics, which is a way we can personalize medicine to study cardiac disease, autism, autoimmunity, and other disorders. We're very basic in nature, so we study molecular biology, which is a way of understanding why we get certain diseases. And we're studying now from pediatric heart disease to inflammation to adult heart failure. We're studying metabolism, which means diabetes and obesity-related research, in the hopes that we can try to understand and find ways to cure this disease, which is devastating the United States. And we're up to date in our technology, so we're doing cutting-edge research that will help target and identify genes or other ailments that may be potential targets for cures and treatments in the future. So the way that our research pipeline works is that we start at the very basic level. So we're interested in identifying how one would get a particular disease. We then translate that into studies that are done in the tissue culture and by genetics, by validating targets and biomarkers using human samples and tissues. We then can do low throughput or high throughput drug screening to identify and generate drugs that might be targetable against these pathways that we've identified as responsible for a particular disease. And then finally, that moves into testing into large animals and clinical trials for validation for mass use in the population. So that brings us to coronavirus. And I know that many of you are probably tired of hearing a lot about what's going on with the coronavirus disease, but I wanted to just give everybody an update if they've been fortunate enough to not be aware of anything going on since March of this year. I know some of us are probably hoping that we're just waking up and it's March 1st and we didn't know anything what was going on. But as many of you know, SARS is a new virus. The first cases were identified in December of last year, so it's almost been a full year since we've been aware of this disease. It probably started in animals. We think it was in the bat, and that then spread to humans. And as this virus is growing, it continues to propagate, and that means it's mutating for its survival. And we're learning more and more in time as more papers are getting published and being distributed for our knowledge worldwide. So how is it spread? We know now that it's being spread by person-to-person contact, in particular through air droplets. So that's mostly from coughing, sneezing, talking, singing, anything that causes air droplets to spew from our person. These can enter your nose, your eyes, and your mouth, and then touching objects that have been contaminated with the virus onto your hands and then touching your eyes, nose, or mouth can also cause the infection. And some people have been thought to be infected following being around people with even having no symptoms. So it's not clear in those cases how that spread may be occurring, but likely there are droplets, aerosolized droplets, that are also contributing to that contamination. So we know now all the symptoms, which, as you all know, our president and many of his staff are now infected this weekend with the virus. It starts with a fever. It becomes a cough, sore throat, fatigue, shortness of breath. Having a sudden loss of smell and taste seems to be a criteria for some patients. Even a headache, muscle aches, and diarrhea. Those are all characteristics of having COVID-19, but they're also characteristics of having any number of different colds and viruses. So you want to be careful, especially now in flu season, that this is not COVID, and so testing or vaccinations for flu are going to be imperative for people. The symptoms start about the first day, between the first day and maybe the 7th day, and can last as long as 14 days. Some people, again, have no symptoms or mild symptoms. And unfortunately, we know that some of these patients, most have predisposition with cardiovascular and pulmonary illnesses, can be sometimes fatal. So I always talk about the prevention of COVID-19 because we've been utilizing these precautions here at our own institute as we've been reopened since June 1st, and fortunate enough to not have had any positive cases here so far. And that's been in large part to protecting ourselves by frequent hand washing, making sure we use soap and water when we can, or alcohol or sanitizer wipes when that's not available. And to prevent transmission from aerosolized drops, we all wear our masks, which I should have. Oh, I have one that says MMRI. I should open that one and show you all. So we all wear our masks all day. Thank you, Nate. And we protect each other because it's really not about ourselves. Wearing a mask protects the next person. So it's a matter of courtesy to make sure that whoever you're around does not get exposed to indirectly to any germs that you might be carrying that you don't want to make anyone else sick. So please wear masks is my only thing about that. In terms of diagnosis and treatment, there are currently new tests, and obviously you all know that vaccinations are currently in the works. The president this weekend was treated with dexamethasone, which is a steroid that has been proven to be effective in sort of suppressing what Sathya is going to talk about in a few minutes, which is the cytokine storm. He was also given a new cocktail of a monoclonal antibody by Regeneron, which is a novel treatment. It binds to the receptor of um, the portion of the virus that actually enters into the cell and helps prevent that virus from entering, infecting those cells. So it is a novel way that they think is proving effective, and it's given probably to the most serious of patients. And the final drug was remdesivir, which is a drug from Gilead, which is thought to be helpful for patients who have severe COVID to prevent the symptoms of those patients for the duration. So that, that triple cocktail, unfortunately, is not something that most of us will get on a first pass. So if you're the president, or if you tell people you're the president, maybe you will get that if you need it when you go into the hospital. But in lieu of that, I think the most important thing is to try to prevent yourself from getting sick and making sure that you get tested if you have any symptoms. So with that, I'd like to turn over to work that is done with one of my colleagues, Dr. Chase Kessinger. And unfortunately, he wasn't able to be here tonight because he has two little ones at home that he is taking care of this evening. But I just wanted to do his work justice and tell you a little bit about what he's doing in terms of preventing COVID. And what I will tell you about MMRI is that we're actually very focused on not studying the disease in its current form today, but we're very interested in thinking about what are the consequences, long-term consequences of individuals who've been infected with this virus, and what will it mean for people down the road in terms of their cardiac function, their lung function, or any other tissues that may have been infected by the virus. And we are becoming very aware now that there are long-term consequences of the virus, even in people who had very mild symptoms of the disease during the initial infection. So we know that they're getting significant fibrosis or scarring of the lung that will have long-term consequences and breathing difficulties down the road, and fatigue. They're getting cardiovascular dysfunction, and Nate is going to talk a little bit about that with some of his work in a few minutes. And they're also getting significant inflammatory responses even months after the infection. So we are learning a lot more as we go, and we're hoping that we will have some solutions for you when the time comes, 2, 3, 4, 5 years down the road. But Chase Kessinger is very focused on an area of research called pulmonary embolism. And this is thromboembolism, which occurs as a consequence of a blood clot that it occurs in the lung or in the leg, and it gets lodged there in the lung, travels from the leg and gets lodged into the lung, causing an embolism. And this is really important because we had an animation here, I'm sorry. So this embolism issue is a really significant problem because it affects more than 10 million people worldwide. It kills more than AIDS, breast cancer, prostate cancer, and motor vehicle crashes combined, but we very rarely hear anything about this disease. It's also a socioeconomic burden to Americans in general because we spend about $16 billion a year treating and diagnosing over 45 billion people who are affected with this every year. So doctors need new tools and strategies to improve the diagnostics, the prognostics, and the therapies for these patients. And now this is becoming a really significant issue with respect to COVID-19 and ways that we can try to figure out how to treat blood clots that are consequent to COVID-19 infections. And so what Chase has been working on doing is figuring out imaging technology. So our patients in the lab are mice. So this is a picture of a heart from a mouse that uses particles that are that have fluorescent tags that can identify where there is blood flow within that tissue. So you can see the blood flow here in the heart from this imaging modality that he's using, as well as here when you can see the blood clots as a consequence of pulmonary embolism and when they dissipate over time. So his work is really important in thinking about COVID-19 because he has a way that he can image what's happening in the lung as a consequence of the infection. And he can figure out ways how the clots might be impacting the infection or the ability of the lung to recover after this infection. So this is an image of a patient over the course of 14 days that has infection with COVID-19. So you can see on day one, there are areas of the lung that are becoming inflamed and clotted with an inflammatory response. By day 5, you can see that the lung capacity is already starting to diminish pretty significantly, because you can see that this accumulation of fluid in the lung is building up. By day 11, that seems to be spreading throughout the lung, and by day 14, you can see that it's starting to dissipate, but now you're developing something called scar tissue or fibrosis in the lung. That means that it isn't healing, it's not healing permanently. So you may be feeling better at this point, but you now have scarred tissue in the lung, which means that over time, you're going to have some inability for lung capacity as a consequence of this infection. And what he's going to be working on is ways to image this and ways to figure out drug treatments that can target that fibrotic area to reduce that scar tissue in those tissues. So I'm going to turn it over now to one of my colleagues, Sathya Unidarthi, who's going to talk a little bit about some of his work on inflammation in the lungs and in the heart. So Sathya, you should be able to share my screen at this point.
Thank you, Maria. Oops. So do I have the control?
You do have control, yep.
All right. Oh yeah, there you go. All right. Thank you, Grandmaster, for giving us this opportunity. And thank you, Maria, for giving me an opportunity to be here. My area of research is specifically in cardiac inflammation, or what happens when the heart is inflamed. And I'm going to briefly talk to you about that and how that relates to COVID-19. So if you look at it, cardiovascular diseases are one of the most common comorbidity associated with COVID-19. What that means is if you take a group of COVID-19 patients and ask the question, what other diseases do these people have? Cardiovascular diseases are at the top of the list. And not just that, people who have cardiovascular diseases and then who contract COVID-19, the mortality rate is the highest compared to other diseases like kidney disease or Alzheimer's. And hyperinflammation or excessive inflammation, like Maria just mentioned, cytokine storm is one of the main reasons or mechanisms that causes this damage. If you look at it, the damage that is directly caused by the virus is not as intense as the damage caused by our body's response to the virus. The body has never seen COVID-19 virus, and the body goes into an extreme inflammatory response, and that is what causes a significant amount of damage we see in patients. So if we want to save COVID-19 patients, we have to figure out how to effectively regulate and control the inflammation. And like Maria mentioned, President Trump was administered dexamethasone. That is primarily to regulate the inflammation in the body. So my research specifically deals with two proteins called TWEAK and FN14. So TWEAK is the ligand and FN14 is the receptor. You can think of it like a lock and a key mechanism, wherein the lock here is FN14 and the key is TWEAK. And when this key comes and binds to the lock, it opens up a lot of things start happening inside the cell. And one of the main important things is inflammation. Inflammation gets turned on when the key comes and binds to the lock. So these lock is actually expressed on the surface of all the cells that we have in the body. And it acts like an antenna wherein it senses the changes in the external environment and changes the cellular gene programming accordingly. And I was interested in understanding what role do these two proteins play in cardiac inflammation. And the way I studied this is that I generated a mutant mouse using molecular biology, wherein we deleted the lock from all the cells in the body. And then we looked at what happens to the heart when we injure the heart. So before I go ahead and share that data with you and tell you how is that related to COVID-19, I want to give you all a brief and a quick Cliff Notes version, if you may, of what happens in the heart when the heart is injured from an inflammation standpoint. So when the heart is injured, multiple different cells start coming into the heart from the blood, And initially, they start repairing the heart from the damage. But if that reaction is not controlled, they go on and start damaging the heart. And two key takeaways from this image is that there are multiple cells that play a role. And all this is like a chain reaction. Once step one starts, there's a highly likelihood that it results in step 2 and step 3 and step 4, eventually resulting in the damage of the heart, unless we stop this chain reaction using drug molecules or some other external means. So in this chain reaction, I was more focused on step number 2, wherein cells called macrophages or monocytes, they enter the heart and then they start damaging the heart further down. And eventually it leads to a phenomenon called fibrosis. So your heart, if you think of it, is a contractile organ. Its job is to contract and pump the blood out. Now imagine if a part of the heart dies and that region is replaced by a stiff tissue that doesn't contract anymore. Now the ability of the heart to contract goes down, and the ability of the heart to pump the blood also goes down. So excessive fibrosis is pretty much replacing that contractile tissue with a stiff region. And excessive fibrosis is not beneficial. So what we see here is that I took a normal mouse, and this is the genetically modified mouse, wherein the FN14 or the log is deleted. And here we are looking at the tissue of the heart. And specifically, I was looking at these cells in the heart. In the heart, when I use a specific stain, these cells appear brown in color as pointed by these arrows. So in a normal mouse, when I injure the heart and look for these cells after 7 days, I do see that a lot of more macrophages are present in the heart, suggesting that the chain reaction has started, and there's a very good chance that we are going to end up in this region. But when I delete my protein of interest, which is FN14, we do see that even after injury, 7 days after injury, we don't see very many brown dots or macrophages in the heart, suggesting that this protein, just by deleting it, we were able to stop that chain reaction. Now, what we see here is pretty much this part of the region wherein we see fibrosis, which is represented by the dark blue region. When we delete this protein, wherein we don't have very many macrophages in step 2, we also see that there is not much fibrosis in the heart after almost 4 weeks, suggesting that a major part of the heart is still contractile tissue and not the stiff blue region. Now, what we see here is actually called a 2D echocardiography, wherein we use sound waves to image the heart. Like I said, heart's job is to contract. And when we apply these sound waves, we see these contractions as waves. And when the heart is damaged, when it's not able to contract as much, we see that the waves are diminished. They pretty much flat out. So we see that in the wild type heart after injury. But if we delete that protein, we see that the heart still has contractile ability after injury as well, suggesting that if we can stop the step 2, wherein we prevent the cells from getting into the heart, we can actually stop this chain reaction and the heart is healthier. Now, how does this relate to COVID-19? So a few months ago, people started looking at the heart sections of patients who passed away because of COVID-19, and were looking at cells in the heart. And just as we saw in the mouse, they saw that a 69-year-old patient who passed away with COVID also had these brown dots, suggesting that the macrophages have infiltrated the heart of this patient. What this means is there's a high likelihood that this chain reaction started in the patient's heart, and there's a good chance that this is going to proceed as well. Not just that, viral particles have also been identified in the cardiac tissue. And recently, there were 2 studies that came out. This study that I'm referring here was carried out in Germany on a set of 100 patients whose average age is around 49 years. And they were monitoring these patients' heart conditions 2 months after their recovery. And a significant people, almost 70 or 78% of them had abnormal cardiac MRI findings, suggesting that there seems to be more fibrosis in their hearts. And almost 60% of them had continued myocardial inflammation inside the heart 2 months after recovery. Actually, another study came out in mid-September. This is from my alma mater, Ohio State University. And they were looking at college athletes. These are students who are at the top of the game, physically very fit. And these people were affected by COVID-19 and they recovered, and they were looking at the hearts of these college athletes. And almost 30% of those really young athletic college students also had cardiac inflammation, suggesting that this is something that might be a long-term effect of COVID-19, and there is a need for us to monitor the patients. These are relatively just 2 studies, and we can draw drawing conclusions from these, but this is just for us to be aware of what might happen. Now, how does the FN14 fit into this research? So we've seen that in mice, when we delete FN14, we can stop this reaction at step 2. So I am testing to see if we can block this FN14 protein in patients, in human patients and patient cells, to see if we can reduce the cardiac inflammation after COVID-19 infection. So we are doing these in vitro or cellular-based studies by blocking the FN14 protein and seeing whether that leads to reduced inflammation in heart. Thank you.
Great, thank you so much, Sathya. So I'm going to try to see if I can, oops. Switch you over to Nate. Nate, I think you should have control.
Perfect. So thank you, Grandmaster. Thank you, Maria. Thank you to everybody who took time out of their evening to attend this. I'm happy to tell you all a little bit about what we're doing, not only for research, but also, helping out our community doing some clinical testing to facilitate operations going on locally. So I'm an assistant professor, so I spent the last decade in Boston, so I'm aware several of you are from that region, whether Massachusetts or Rhode Island tonight. So I spent quite a bit of time there training at Massachusetts General Hospital, Harvard Medical School, and the Broad Institute, and joined the Masonic Medical Research Institute, and effective March 1st of this year, I believe. So first part of this quick 2-part talk, a little bit about COVID-19 clinical testing here at the MMRI. I think this is a good story, to highlight the flexibility I think that our institute has to take on a challenge that comes on, really out of left field, and our commitment to be good members of the community. So I think we really touched on both of those things within this really short component of my talk. So a little bit about the statistics. I'm mostly saying these. I know, like Maria said, we've seen a lot of this stuff again and again over the past several months. But what I really want to say is some of what we've done here in context of these numbers. So these are the nationwide metrics. So from early April through to now, we've seen a great increase in daily testing of different modalities that I'll talk about in a second. Case numbers. So this spike here was the original one centered in the Northeast. This larger spike here was the outbreak in the Southeast and Southwest in the last, going through the last 2 months. Currently hospitalized numbers, I guess some amount of this lower case number versus hospitalized number are probably due to 2 things. One is increased test volume, as here on the left, and the other is the demographics of the people that were affected. So the second spike in the South was a lot of younger people. So I guess the death versus case number ratio kind of changes based upon those demographics. So here it is in the Northeast. It looks, I guess, a little better with, our test numbers continually increased. We had that really large peak right in the beginning of the outbreak in the US, held steady through the summertime. And then I think we saw what we would consider an expected uptick in case numbers, and a slight bit in those hospitalized, likely due to, colleges reopening and schools now reopening. So I think a lot of that stuff's in flux now. And, we're certainly doing our part to test here to help out to make sure that schools and everything can remain open. All right, so just to pull up some numbers in New York State, just again, this is for context. I'm going to go through this, actually what we did according to this timeline. So very early that we were hit on. You can see the number of tests is in blue here. So, through April, really up until, mid-June, these daily test numbers were really low, and the case rate, which is in yellow here, was relatively high. So I think I correct me when our exact date of shutdown was, Maria, but I think it was, maybe mid-March. I think I was about ready to move.
Yeah.
And Maria and the administration made the wise decision to shut the facility down, and then we're able to reopen safely. I think, it was best case scenario for the way that we handled it, and we're able to open up and resume our work. So in New York, we maintain this kind of low relative percentage of infection, which is this orange line here, and our tests are hanging around steady. So what we wanted to do here is, let me step back just for 1 second. So this is when we were making a lot of our decisions, and I think our Maria's mission and our all mission, not only as people at the MMRI, but citizens in the Mohawk Valley and citizens in New York State, and I guess just citizens in general, wanted to say, what can we do? And the state was having a tough time getting these blue lines or the test numbers up, and especially for the Mohawk Valley. So I guess I'm a bit of a believer of local solutions to local problems often work. You see the people that you're going to be working with. You lack a lot of the large institutional problems when you need really big groups of people to make decisions. Perhaps it's a little more difficult than these one-on-one things. So our local community hospital here in Utica is the Mohawk Valley Health System, and we decided to form a community-based network with them, operating with their clinical laboratory to facilitate their testing to do what's really a local solution to our local COVID problem here in the Mohawk Valley. All right, so one quick diversion back to this. So this was New York State. This was what the Mohawk Valley looked like. So if you can look for the trajectory of these test numbers, the Mohawk Valley was doing pretty rough. We had, relatively high case number. These were all very, very symptomatic people that were being tested at this time in these very limited numbers. And what we wanted to do is around this time, come together and make a plan for what we were going to do to help. So for that, we formed the COVID-19 task force within here. I think it was not only for the testing, the way that we're going to handle getting safely back to work or working remotely for the time being in early April. We reached out to New York State very soon thereafter to say, we had the capability to do testing and to help, the state with these problems. Admittedly, the regulatory barriers for a lot of this stuff were frustrating. So we had the ability, and getting through to actually help was a bit more difficult. 1 second here. We then had first discussions thereafter. So, one thankful thing I am for the state is they gave us options. We said we really wanted to help. What can we do? And they suggested that we linked up with hospitals so that we could, work as a team to actually facilitate this testing. So I think it was the beginning of May that we had our first discussions. And by June 1st, not there long thereafter, we had identified a test that would work on our equipment. We had validated that test in-house, and we're starting to run tests for the local hospital. We agreed when we were ramping. We said, let's ramp up and do a light volume that first day, and they dumped 350 samples on us that first day. I think I was here until 11:45 that night trying to figure out how to get everything reported back. But within 2 weeks thereafter, our turnaround time, which at the time was horrific around the country, we returned everything in under 24 hours. Midway through July, we'd hit 5,000 tests, and I think a couple weeks ago we hit 10,000. I think we're just over 13,000 tests that we've run here at our facility. There are some, we're not Quest Diagnostics or LabCorp or something, but we certainly meet the needs of our local hospital. They've been very happy with our turnaround time, as well as a attention to detail that I don't think that you necessarily get out of a lot of these large testing labs. So I say, when we're working with them, and perhaps many of you are aware with some of the issues with particular tests and false positives and false negatives. So, a lot of these assays are things that I've done for 15 years through my research career, and so have the folks in the lab performing the test as well. So when we see a result, we go back and we look at the machine and actually confirm it. We didn't want to give someone either a false positive or false negative diagnosis. All right, so a quick diversion into the three types of tests that you may hear about now. So molecular test. The virus itself has a genome, much like us, although the sequence is different. So that allows us to use this test to detect the virus. These are super, super sensitive tests. There was some risk of false positive, which is why we always went back in our hands and actually, looked at the results manually for anybody who tested positive and make sure that was the case. These ones will molecular tests say if you're actively infected right now. Antibody tests, they look for antibodies within your body that would recognize the virus. So those detect whether or not your body has seen and had a reaction to the virus in the past. They use those, I think, mostly for modeling of, the percentage of the population that might have been previously infected. I don't think they're an effective solution for any kind of, you don't have to take, modifications of your behavior anymore if you have a positive antibody test. So the third, which I think has gotten a lot of news lately, is an antigen test. So instead of looking at the genes of the virus, as here on the left, you actually look for the proteins, so the stuff on the outside of the virus that it exposes. And the antigen tests say, basically through this little what we call a sandwich ELISA here, says that yes, there's a piece of a virus protein in this given sample. You can think of it as similar to a pregnancy test. I think is the surrogate that most people would be familiar with. So really sensitive, has some possibilities for false positive if they're read incorrectly. Generally with antigen tests, your problem are what we call false negative. So they aren't as sensitive as these. So there's a possibility, the most common failure is that someone has a virus, but only a small amount and not enough to reach a threshold for that test. So they're actually positive, but called negative by the test. So we run molecular tests here at the MMRI, and I went downstairs and took pictures of the pieces of equipment here. These equipment that make this testing possible and has made that whole program possible, I think, is due in large part to the generosity of the Masonic community. After I moved here, this place is incredibly well outfitted for any research enterprises that we want to do. And, having machines like these, so these are two real-time PCR machines. This is an extraction machine to actually get the genomes out of the virus. Having these machines really makes it so there's no limit on the flexibility that we have, or the type of experiments that we can do. So I'll say personally, at least, I'm really thankful for the generosity of you all for helping us have the operation that we have here. All right, so just run through numbers first. So since June 1st, that's a little dated now. I didn't include the last few days worth, but a little under 12,000 tests. Our turnaround time is well under 10 hours. If we get the sample from the hospital by noon, they'll have that one back by 4 o'clock. So I guess our rough turnaround time is if we receive our ones at 5 o'clock, you'll get them the next morning. I wanted to say these are the people that have done it. So if you ever do visit and see Ryan Pfeiffer, Mayuri Desai floating around the building, I hope you take the opportunity to thank them for the hard work that they've done for this. So I'm more of the facilitator, and these are the two that are actually doing all this work. So when you look at this number, that is these two people. Things that this helps, when we started this up, we were just going into phase 1. The capacity that we were able to give was part of what allowed Central New York to originally open. Helping patients go home. There's a requirement for nursing home patients to receive several negative tests before they're discharged to their living facility. So our fast turnaround and flexibility helps patients go where they want to be. Surgeries and outpatient procedures, so the normal things that we all need to get done from time to time require a negative COVID test. So a lot of the samples we're actually doing now are people who want to have the, in normal times, what would be a no big deal thing now require a COVID test. We turn those back around them often the same day. And then we're also, I'm not going to go into the data, but we partner with the New York State Department of Health. So they receive all of our positive samples, help track the way the virus is, not only moving around the state, but also between facilities. So, for example, looking at various nursing homes around the area when we had those really unfortunate outbreaks earlier in the year, and trying to discover how that's transmitting between the different places. So we continue to work with them throughout time to, help understand the way that the virus is moving and if there's any ways we can stop it. So that's it for that section. I wanted to break into just a, hopefully a relatively brief conversation on some of the research that I do. So my normal career is not running molecular tests for any given virus. I was trained as a geneticist and mostly focusing on cardiovascular disease, but also I think somewhat of a technologist. So my research really pushes the bounds of what we can do with some of the things that have only come around in the last couple of years. So one particular example was to apply some of what we've been learning to cardiac specific complications of this disease. So this is a really small part of a large research program that we have going on that, when the pandemic came about, we really said, here's something we can deliver on really quickly using unique technology. All right, so much like's been mentioned previously, around 20 or 25% of the people who pass from COVID-19 infection has what we call cardiac involvement, meaning there's direct damage to the heart. But questions remain, and Sathya talks about some components. There are some open questions here, but how is it being affected? Is it being infected directly? Remains an open question. Who's going to be at higher risk for cardiac symptoms or for the direct infection of the heart, and why might that be the case? And I think many of us are on what are common medications, and one that was of a large concern in the beginning was antihypertensions or high blood pressure medications colloquially, and there was some concern that they would actually increase the risk for tissues being infected by COVID itself. So what do we do? In the past, when we studied a lot of molecular things, so this is a sketch of the human heart. We treat the heart as a thing, but that heart is not a single entity, but is instead made up of many things. So I'm gonna divert into I guess more appetizing analogy than the heart. But you can imagine, cookie doughs or cookies themselves are made up of many different components come together, and you'd say this cookie is good or this cookie is not. But to understand why it's good or why it's not, you need to go back to the components. So, understand if you had more sugar or more flour or the like, or you want to know is the quality of a given ingredient different, and that's what made that cookie that given way. So to go back to the heart, the equivalent of the ingredients in your cookie batter would be all the different cell types, much like Sathya alluded to, that make up this heart. So we think there's at least 10 or 12 different types of cells or different ingredients that make up your heart, and all need to work together. We think of it as a pump. So often people focus on the one that does the pumping. It needs a lot of support to do its job, and we have to understand what all of them are doing. So how do we do this? Hopefully this video works halfway decent. It's always choppy through Zoom. So what we have are these things. It's an engineering solution to go through this problem. So this is what we call a microfluidic device. So it's about maybe the size of my pinky fingernail. It's the size of the whole device. And you get these little channels that run through here that are 100 microns. So think of a millimeter, and then it's 1/10 of that. And through this, we can break the heart apart into its components, pass them through this device, and actually label each one of these components individually and learn a lot about it. The way this ends up looking is this large map. So it's fuzzy because there's so many individual points of data here. But each of these numbers would represent a different type, a different ingredient in what would have been your heart. And each one of these points is somewhere between 1,000 and 10,000 pieces of data. So you can imagine this is I think we're about 300,000 individual cells. So somewhere around a billion or a billion and a half individual data points. So from that, we can look at those questions that I mentioned initially, and only in this resolution of looking at each individual ingredient or component of the heart, and learn several things that were really important. I think for guiding clinical care, for guiding treatments, and hopefully understanding the disease in general. So the first thing, the place most likely to be infected in the heart are these cells that line the smallest blood vessels of the heart. So I think we're all familiar with perhaps coronary arteries that are most likely to get occluded to form a heart attack. These are the smallest pieces that branch off that supply all the pumping apparatus of your heart. Those are the ones most likely to get hit by the virus. People with pre-existing heart conditions, in our case, what we call cardiomyopathies or going towards heart failure, have more of what we call these receptors, things that can bind and bring the virus in, than people without the pre-existing conditions. So I think, as we all understand now, people with these conditions need to take more care with their health. And then finally, I think it was important, and part of a lot of the guiding literature in clinical space is that we looked at the receptor, which is a surrogate for its ability to be infected, and said all these medications that millions and millions of people are, concretely do not have an effect on the ability of the virus itself to be taken up by the heart. So we were really thankful to be able to provide that information. So if we go back to our timeline here, just to show when these things happen, so that we're doing research in the midst of what was a shutdown. So this was a large paper that was, published, I think, online on the 18th of May, was released officially in print in mid-June. So really a survey of all the different cells or components of the heart, published in the premier cardiology journal, I think, in the world. And then specifically the one related to COVID-based infection, got a fast release. So, pandemic starts here. A month later, we released a paper saying all the conclusions that we just did. That was released in what we call preprint. So, not peer review, but just, it's important we get the information out there as fast as possible, and then officially released in print in late August. So, plan moving forward, really briefly, we continue to monitor outbreaks both locally and in concert with the state. Flu testing is going to be a big deal. So like Maria said, everyone, please get your flu shot. It certainly complicates our infection or our testing protocol. So this is by weeks in the year. So this would be January. And this graph says, influenza positive rates from the flu. So we are right here. In the next few months, we're going to see this ramp up. And we're working to implement an assay that can do all of those, to continue to make sure that people are staying safe and correctly diagnosed and treated appropriately. But I'm really hopeful that this flu rate thing will go down for all of us in comparison to normal seasons. But we'll be working with the hospital, help them out as well. We're working using that same single cell tech to look for the virus directly in cardiac tissue. Is it getting in there? That's in partnership with Emily Tsai, who's a heart failure doc in Columbia University. So we imported a bunch of human samples to work on to say concretely what the virus is doing in the heart. And then finally, the sad note is, when we're working on these projects, whether it's the clinical testing and getting the best logistics to do these, or our particular research projects, SARS-CoV-2 was built, or our understanding of SARS-CoV-2, which is COVID-19, is built upon the experience for SARS-CoV-1, which was the original SARS back in the early aughts, mostly restricted to East Asia, the really severe outbreak in Toronto. So on the basis of things that we understood molecularly, the way it spread was derived from that virus. And in the unfortunate, but probably inevitable scenario that SARS-CoV-3 comes around, hopefully we'll be more ready to tackle that in all aspects. So, Maria,
Right.
thank you.
Thank you so much, Nate. Let me see if I can try to take control again. Well, I can try to do it this way. So I know that many of us have heard a lot about COVID and all the things that are going on, but is there life beyond COVID? And really, are there other things that we're thinking about with respect to other diseases that may be more important and prevalent after the COVID situation? And what I want to point out is that the Masonic Medical Research Institute was really founded on its attributes with respect to cardiovascular disease. And cardiovascular disease, COVID notwithstanding, remains the number one cause of death in the world. And if you were to combine it together with the incidence of diabetes and obesity, which actually has become profoundly elevated in the last couple of decades, we have over 500 million people affected by cardiovascular associated illnesses in the world, which can become a huge socioeconomic burden for us in the future. And you can imagine, I just swiped this off of the internet, but you can see why this can become a significant problem for people. This is just a fun, I like this slide because it shows people wanting to be exercise conscious, and in order to do that, they take the escalator up into the fitness center, which I think defeats the whole purpose of getting exercise in the first place. But really, when we think about what happens with respect to the obesity epidemic, you can see why this is becoming such a significant problem. You have, over the last 25 years, gone from a country that had less than 10% obesity rate to now having, from 1985, to now having virtually 9 states, and this is a little outdated in 2014, but you can appreciate all the red in the colors showing that we have obesity rates that are elevated above the 30th percentile, and 9 of those states, as of 2019, are actually over 35% increase in obesity. 35% individuals are considered obese, which is an extraordinary number of people who have this problem. So if you look at that map now from 2014 and extrapolate it to this map, which looks like the same map, you will be surprised to note that the same incidence of individuals who are elevated in obesity are also now dying of heart disease and diagnosed with heart failure. So this is actually a map of individuals diagnosed with heart failure. So it maps almost perfectly with the map in diabetes and obesity, really kind of emphasizing the significant role that this will have on our lifestyles and our life longevity in the next few years. And in fact, what I will tell you is that for the first time in several decades, probably since 1950s, the lifespan expectancy is thought to be shortened somewhat, in large part because people are suffering from this obesity epidemic that leads to earlier incidence of cardiovascular disease. So we're very interested in trying to figure out how to, let me see, how do I take this back? I don't think I can take it back, but I'm going to try to keep going. So what are we doing about it? So as Nate discussed, we're all interested in focusing on the heart. We break down the heart into several components, cell types that he mentioned, like the cookie recipe that he discussed, but you can break it down even further and study each cell's genetic code, and that's at the level of DNA, which you can appreciate here on the right. And by doing that and studying individuals' genetics, we have a capacity to really study all kinds of diseases, including diabetes and obesity, which I'm highlighting work from Adia Romalaran, who was recruited recently from SUNY Downstate. We're working on genetics and heart disease with Zi-Chang Lin, who was recruited from Children's Hospital at Harvard University. I'm also studying pediatric heart disease and genetic disorders, and I also came from Boston. I was at Harvard Medical School for 15 years. We're studying inflammation, as you heard from Sathya Unidarthi just a few minutes ago, cardiomyopathies and epigenetics, which are things like stress that impact the heart and cause heart disease as a consequence of things like that. We're studying heart failure, which you heard recently from Nate Tucker, as well as cardiac arrhythmias, which are associated with sudden cardiac death. And I'm highlighting Gary Astrup and John Cordero, who I don't know if you guys can see, he's actually behind our pictures here on my screen. So much of the work that we do is also involved in drug therapy. And Dr. Dr. Jason McCarthy, who came from MGH, also in Boston and Harvard Medical School, is very interested in nanoparticle drug delivery. So it's really a platform, it's a drug-based moiety that encapsulates drugs to deliver them specifically to individual cell types so that you can deposit the drug directly into those cell types. And by doing that, you can actually prevent off-target effects, increase efficacy to targeting that disease where it needs to be, and not have all of the side effects like chemotherapy drugs might have on skin and hair and gastrointestinal tract. So in addition to our cardiovascular work, we're very interested in other areas of science. We're doing neurodegenerative diseases like autism and Alzheimer's. We're doing autoimmune diseases like systemic lupus, erythematosus, rheumatoid arthritis, and diabetes, gastrointestinal diseases like Crohn's and gastric cancer. And we're very interested in moving into platforms. In fact, I have a project that's just starting on glioblastomas in the brain. So I'm very excited about some of the work that's continuing to be ongoing. And with respect to that, we're also doing a lot of novel technology work. So many of you have heard probably of inducible pluripotent stem cells and using genome editing through CRISPR-Cas9 to correct the genome for that mutation so that we can eventually treat those diseases at the level of the DNA. We're using gene therapy as an approach, which uses viruses to deliver drug much in the same way that I talked about nanotechnology. And we're using other drug delivery targeting platforms to be able to treat those diseases based on the mechanism of discovery for how they're caused. And with respect to the neurodegenerative diseases, I just want to highlight some work where I was fortunate to recruit a young investigator from Yale University who is a neuroscientist and does a lot of work on autism. And so her and my interest with respect to pediatric disease actually came together in a very fruitful way because we had a hypothesis that we thought that the cardiovascular genetic diseases that are caused in pediatric patients may be mechanistically exactly the same as the as the genes that cause autism in patients. And so we've combined our efforts to actually do inducible pluripotent stem cell research where we can take a somatic cell, which is a cheek cell, a cheek swab, or a blood sample from any one of you, and we can turn that into any cell type of interest that we want to study. So what you can appreciate here is in this particular slide, I'm showing you a patient's blood sample that is now turned into the brain. And you can see actually the development of the two eye sockets in the dish from this particular patient. And we can also take the same cells and turn them into that patient's heart. And by doing that, we can actually study how the genes involved in a particular disease evolve differently in that particular patient, and then use drug treatment therapy to understand what drugs may be most effective in that particular patient. So we're in the process of doing a lot of this work now to try to understand how autism occurs and whether or not it's related to cardiovascular disease, as well as some other projects that she's currently working on with respect to autism. And that entire mission actually has been in large part to one of our directors, Patton Bimbo, who's been such a huge proponent for our autism-based research. And so with that, I'd like to end and hopefully have some time for a couple of questions if anyone is interested. And I like to end with a quote because I actually just saw a biography on Marie Curie, and she's become my mentor as to come full circle to Grand Master's opening. And she had once said that nothing in life is to be feared. It is only to be understood. And now is the time to understand more so that we can fear less. And I think through research and the work that we're doing here at MMRI, we're actually creating opportunities to understand why people get disease so that we don't fear them and we can figure out ways to resolve them and evolve and move on. So our success metrics have really been all the things that we've talked about, is to improve our quality of science, to succeed in getting our grants and funding, profitability in our operations, and that's in large part thanks to all of our contributors and our donors, our scientific advisory board, establishing relationships, which we've already well done with hospitals and academia, and then having seminar series that allows us to keep, even though we're in upstate New York in a small community, keeps us abreast of what's going on around the country and around the world. So we're building, we're continuing to grow. We hope to continue finishing our construction. We want to increase our scientific investigators to continue that growth trajectory. We hope to increase our research scope. So we're already moving into cancer. We've already evolved into immunology. We want to take on more bioengineering and artificial intelligence as part of our work. I think robotics is going to be the wave of the future in terms of diagnostics. And I think I mentioned to the Grandmaster just recently that I read an article last week that actually is using artificial intelligence now to detect through the retina whether or not one is infected with COVID-19. It's not quite perfected because the sort of percent accuracy is about 70% in that, but they detected about 90% of the individuals who are infected with something. So at least it's moving in the right direction. So it does seem to have something behind your eye that can detect whether or not you have some type of illness. And our long-term goals, obviously with much of your help, is to increase our research facility capacity, to create startup venture based on our ideas and our technology, and to be able to develop incubators that can continue our growth. And if you're interested in finding more about us and anybody who works here in their research, please feel free to look at our website at mmri.edu. So with that, I'm going to turn it back over to you, Grandmaster, and I'm happy to take any questions.
This is terrific. You know, I had the pleasure as Grandmaster, even before I was Grandmaster, to walk the halls of the Institute. And I got to tell you, I have a hard time hanging on to the conversations to try and understand it. But when Nate brought it back down into cookie dough level, my one-cylinder mind hung right in there, and I got it. So I appreciate that ability to bring it into to my level of understanding here. I did have a couple of questions that came up here. We have a Grandmaster from the Northeast who's probably very disappointed that all of these Boston people are here now in another location, but that's where they all started. He says when speaking, he is at least 10 feet from anyone and typically removes his mask to improve his speech quality. Is this a safe practice?
So I can take this easy one. Well, it's not so easy though. So I think in general, we have been taking our masks off if we're at least 6 feet from one another. I just saw an article that just came out yesterday to suggest that perhaps that may not be effective enough, and that even within 6 feet, you still have the capacity to spread the aerosolized COVID particles. Moreover, it seems like these particles actually stay in the air for a lot longer than what was originally anticipated. So even if someone is infected and exits the room several minutes or even an hour, someone else entering that room can still inhale those particles and become infected. So it's still a work in progress. I think in general that is a safe practice. It has seemed to work effectively for us, at least, and it has seemed to decrease the rate of incidence of number of individuals that end up being hospitalized as a consequence of the disease. So I think it is good practice, but I think be cognizant that there is new research coming forward to suggest that maybe that there may be new protocols put into place depending on what we discover about how long things stay in the air and whether or not things project farther than 6 feet.
Okay. High blood pressure, a heart disease by itself, or is it a condition which, if untreated, leads to heart disease?
Sathya, would you like to take that one?
Sure. I would call hypertension more of a cardiovascular disease rather than just a heart disease, because there are multiple other components that come into the picture before hypertension is established in a patient. And the second question, like, would untreated hypertension cause heart disease? Is that the question, Grandmaster?
If untreated, does it lead to heart disease?
Yes, if hypertension on a chronic basis, if it's untreated, it can lead to heart disease. That is correct.
Okay.
it actually is called the silent killer. So it is something I think that people do need to make sure that they monitor. If they do have high blood pressure, it's important that they get medication to properly control.
From Cape Town, South Africa, we're all over the world here with what you're doing here. Edgar Barron wants to know that if this can be taken globally for what you do. He has a question that he put in here. Does the MMRI make research available to Grand Lodges around the world, and is there a process for this?
Grandmaster, that's your question.
All right, it's a deal. It's a deal. I can do that one with no problem at all. In fact, if anybody wants to email me, I can put them right in touch with Anthony Cucci or somebody else in the location there that can make this all work. You know, there was a time where we were thinking nothing of getting on a plane and going and speaking about this anywhere, but you know, now almost every state in the United States is on the naughty list, and nobody wants to go on planes, countries, and the quarantine times. But we are still available through this medium, and we can still do what we need to do. The research that's done at the MMRI is a global aspect of it. It's not a New York aspect of it. It just happens to be in New York. It's got to be somewhere, and it just happens to be in New York at our Masonic Care community. But what they do benefits us, our children, our children's children. And that's the way I look at everything that we do. So when it comes to raising money and when it comes to supporting it, that is the way I look at it. You know, we have a lot of things to raise money for. I get it. And I'm running a telethon to try and raise more money. But, you know, we can take this on a global thing. So Edgar, no problem at all. If you want to get a hold of me, I will link you up with the right people. And they can be at your doorstep by Zoom or some other virtual means to do this. COVID-19 will go away and be defeated. Dr. Tucker mentioned the previous COVID with a hotspot in Toronto. Is that an active COVID still affecting people?
Well, so SARS-CoV-1 was eradicated, I believe, 2005. So it's just similar in strain to the particular COVID. So these things exist, you know, mostly in animal hosts. All the time, and every now and again they jump to people, and both SARS-CoV-1 and SARS-CoV-2, and probably when SARS-CoV-3 comes around, that'll be from a similar mechanism. So SARS-CoV-1 is dead and gone. SARS-CoV-2 will go away with vaccination and good behavior and time, and we'll talk about that in the past tense as well.
Good. There was a question when number 3 was coming, but we don't know when number 2 is gone, so it's hard to say when number 3 comes. Do young scientists get to pursue their own ideas at the MMRI? Well, you all look kind of young to me, so I'm guessing that you kind of do. So
you're as old as you feel.
In that case,
I feel old today. Yeah.
In that case, I feel terrific. So I guess you can pursue. Actually, I guess you're brought to the MMRI based on what your dreams are, your research is, and the end result of what that research could be.
Yeah, you might take part of this, Maria, because I think I'm kind of in the so there's a few different stages of young here. So there are young faculty members like myself and Sathya that are just starting a research program.
And me.
Yeah, and Maria. And, so you know, getting the opportunity to really, you know, you've spent all these years training and to find your own direction and go. So yes, in that respect, and we're supported really well by the Institute to really take those chances and do what we think is important. And then there's the people that are, you know, a few years behind me that we're training here at the Institute as well. And I say, at least personally, I'm not smart enough to come up with every research avenue that we need to go, and only through training the next generation of people who sit in my chair here, and Maria's chair, and Sathya's as well, do we really get the most benefit about what we're doing. So I always, and I think both of my colleagues here too, you know, you want people to have the space to take their ideas somewhere while also, you know, training them how to progress their career at the same time. So I'd say yes and yes and yes.
There's a question. Yeah, go ahead.
I was just going to say I agree. And when I was a postdoctoral fellow, I was fortunate enough to work with one of my biggest role models, which was Lou Cantley, who discovered an enzyme that I've worked on for my entire scientific career, PI3 kinase, if anyone's interested. But in any case, he asked me one time during lunch, he said, if you had all the money in the world, what would you do as a postdoc? So, I was sitting with a group of other postdocs, and I said, I would retire, I would travel, and I thought, and I think I said, and I look back at it now, and I wonder how prolific it was, because I said I would actually love to run a research institute where I have basically the ability to watch all kinds of different research really blossom and grow. Of course, I said it would be somewhere in the Caribbean, so that backfired. But the rest of it, I think, being here at the Masonic Medical Research Institute really kind of came true. And I think as you grow and your research develops, you have a handle on what you've loved and what you've developed as a scientist. But as your career grows, having this opportunity that I now have, which I have loved as part of this new job, is to watch everyone else's career kind of come to the forefront and really rise to the top, is actually, it's almost like having grandchildren in a way, and it's very, very exciting and encouraging. And so it just keeps the science going. We can't obviously answer every question that comes around, but the data drive the questions. And so you follow the data and your research just continues to grow.
Great. You have a statistician on staff, and how do you handle statistics of research?
Yeah, so I'm going to give that to Nate, but I see that that's from George Romeo, and I just want to make a shout out because he has sent us, I think, every single paper he's published so far in statistics on COVID. So I just want to say thank you for doing that. But also, I'm going to turn it over to Nate because that's an important question, and we are addressing that.
Yeah, so there's a few different levels of statistics, and I guess fortunately or unfortunately, when you get to the data science space that I am, that the statistics become very complicated, and you need to, either work directly with statisticians or become proficient in what you need for the analysis of your given problem. So a lot of the stuff that happens in the lab is pretty simple statistically that we can handle here. As well as development of some of the stuff I'm doing in my cookie analogy there that I and my group handle. But I guess the answer, someone with the title statistician, would be no. But definitely work with external statisticians as well as pretty extensive statistical expertise within at least my group.
And I just want to add, some of us still have our affiliations at Harvard, and so we are jointly bound to the Harvard Catalyst program, which provides a full statistical analysis guru group that if we ever needed, we could go to as well.
I think we kind of poured through some of the questions here. I mean, what can we do to prevent the spread of SARS-3? I guess we got to get rid of SARS-2 first, and then we can figure what it is. But I'm betting you that my $9.99 masks that I'm selling at the Grand Lodge will probably be helpful to prevent the next spread of anything that comes out. Profits of which go to the charity, obviously. But I guess that would be, how do you prevent the development of spread of SARS-3? I don't know. We also had a question, do we know if it came from a Chinese lab, SARS-2?
I don't think that there's any evidence for this being a constructed or manipulated virus at this time.
And with certainty, actually, so the entire genetic code of the virus has been sequenced, and it is a naturally occurring virus.
Okay.
And like the other SARS viruses, it's likely jumped from an animal to humans.
Extra heart deaths occurred during COVID? I would imagine that there probably were.
These are complicated questions because it's often, people with severe heart disease who are then infected by COVID that really have multiple issues going on that eventually passed. So having a designation for cause of death that's exclusive is, a little more complicated. We'll have to look at these big, which I think gets to your preventing the development of SARS-CoV-3 or timeline, or all the, what we're going to do over the next few years is what we call a really intensive retrospective effort on, what was good, what wasn't good, what could we possibly do, and what the symptoms were of those people, how the virus acted, and through all of that, hopefully, we'll do a lot better next time. We can, maybe make it so it never jumps, or we keep it restricted to a given location, or treat our patients better when they do become infected.
Here's a good question. What type of equipment are you looking forward to get to help your research? I know what you want is not on Amazon. So this is
Might be.
maybe.
Yeah, it'd be a big item. Yeah.
Well, I think one of the sort of big ticket items that I think we're hoping to get is a new sequencer, an Illumina sequencer, which will allow in particular Nate, but also Sathya and Gulhan and others who are working on genetic sequencing to get information that they need to continue their research efforts. So that's something that I think we're hoping to get in the near future.
How much is a sequencer?
Depends on the model.
Well, Nate actually did a few proposals for me recently. So Nate.
Okay. I mean, let's if we're gonna throw this out, let's
Was it 450?
let's go for the sale. Let's go for the close of the sale. How much is it?
450. Yeah. Sorry.
Around $450,000 will get us a state-of-the-art sequencer that really fits the needs of the scale of the institute at this time. Right now, we're at the whim of outsourcing. So I send my samples to New Jersey or Boston. They're sequenced there, and we have the expertise to run this equipment here. We've had other sequencers in the past of various kinds. When that thing is in your hand, I can do my experiment or Maria's experiment or Sathya's or whoever else needs it today. I don't have to wait 2 weeks. The cost will also be, comparable, if not better, than our outsourcing as well. I think it's something that could bring continue to bring the institute on the upward trend of research that we've been on for some time.
Okay. I think, are you a 501c3?
Yes, we are.
So that means that if there's a donation made to you, it's a charitable one.
Yes, it is.
Okay. So
Wow.
if there's any eyes lighting up out there, there's an opportunity to help humanity with this. So a little plug there for you, because I know that you guys are shy when it comes to asking these things, but I'm not shy when it comes to asking for the sale. The more heart and other medical problems a person has means more mask wearing, distancing, etc. I would imagine so. I guess maybe not even going out and doing things for the time being if you have more medical problems.
Yes. So is the question, if they have medical conditions, they shouldn't go out?
The question is the more heart and other medical problems a person has means more mask wearing, distancing, etc. And I guess, are there more things you need to do?
Yes, encourage everyone around you to wear a mask, because wearing a mask alone is not enough. It's the other people spreading it to you that is the problem. So make sure that you're wearing a mask around you.
Well, I think we've gone through the questions here. I know everybody has lives. I could do this all night. I know that there's a grandmaster out there who is, I've seen that he has gone around and speaking at his lodges. I haven't done that. What I've done is I've shut everybody down. So in order to, first of all, to keep the number of individuals at the social distance locations down. And also, we don't want to obviously bring somebody in that might have it. And this hurts me because I like to talk. So what are you going to do? I have to just do it on my own. We have one here from Lebanon. God bless you, Nathan, Sathya, and all the scientists who work with you. Thank you for the awesome work you are doing. Rafi Timoni. He's the Grand Master of the Grand Lodge of Lebanon. And I just got a nice email from Nadia, who's a doctor in Bulgaria. It's so nice to know that you're helping such useful institutions as Medical Institute that makes research useful for the humanity. So we had a global reach today, as we always do in Masonry, and I want to thank all of you for joining me. I know that some have asked if we can give them the recordings of this that they can put on their social media to use. So I'll work with Anthony and Dr. Contaridis to see how you want to do that, if we want to edit it, things of that nature. But in the meantime, I thank you all for what you're doing. I'm a lucky man to be a Mason at this particular period of time and place because we are learning more today than we did yesterday. So thank you again. Once again, the Masonic Medical Research Institute is a global institution. It's sitting somewhere, but it has a global reach, and all of you can participate. Thank you again, everyone. Have a safe night. Have a safe life.
Thank you.
Thank you.
Thank you.
Thank you for your interest in our cutting-edge biomedical research efforts. If you would like more information about the Masonic Medical Research Institute, please visit our website at www.mmri.edu. If you'd like to get in contact with me personally, my information is listed at the bottom of the screen. Thank you.